David Nguyen and I, we met in Mary Helen's lab when I was already, I think, I was a project scientist. And like I said, 2005 is when he graduated, and we were really much involved in trying to. I was really fascinated by David coming from the biology. Had a very strong emphasis actually on quantification. It was very precise, and so we really got a good relationship in terms of trying to work together in terms of quantifying immunocytochemistry and immunohistochemistry data that he would acquire for his PhD with Mary Helen. And so that was a great experience. And then I moved on to my own lab, and David, when there was the move to NYU, David moved along with Mary Helen, and I stayed behind. So, life kind of like came apart, but I would say that David was really very much instrumental in a lot of the thought process behind the research that was done with Mary Helen. I remember these conversation between David, me, and Mary Helen, where we really were trying to understand better how radiation affects tissue, and David was bringing a new perspective, more on the tissue side that I didn't, that I really enjoy. So I'm sure David is going to talk a bit about this kind of quantification and other kind of thing along this line. So thank you, David. It was a pleasure to work with you.
All right. Well, thank you, Sylvain. And I just want to say that this is a theme that will arise several times today in terms of Sylvain's influence on people in Mary Helen's lab. I started in the lab as a biologist, and of course, Sylvain being a math and physicist person. After interacting with him, I went from saying the coffee creamer is very sweet to saying the coffee creamer sweetness is 9.561 on the scale of 10. So that's the effect that Sylvain has on people. All right, so let me share screen. All right, and then let me do this presentation mode. Okay, right. Okay, people see my screen? Yep. All right. Well, when I first joined Mary Helen's lab, I was a molecular biologist. Love cell culture, molecular cloning, things like that. When I joined the lab, she gave me the choice of working on a DNA repair project that was involved again molecular cloning, or to do a mouse model tumor biology project, which to me at the time was very foreign, and being a risk taker, and believing that with great risk comes great reward, I decided to do this tumor biology project. I mean, what could go wrong, right? Yeah. And so I joined the lab to do a tumor biology project under Mary Helen's guidance. It was this project, as Sylvain mentioned earlier, was just a really very clever, very brilliant experimental design that allowed us to test the effect of radiation, how radiation promotes breast cancer without ever touching the cancer itself, right? Which was very innovative. And here's a quick infographic. On the left-hand side, the experimental setup is essentially to remove the naturally occurring mammary gland in the growing mouse, such that you have just empty fat pad, which allows you to then irradiate the whole mouse such that the whole body has been exposed to radiation. But then you can transplant in the mammary fragment from a donor mouse, right, which has never been irradiated. Okay, and this donor mouse will grow to form this donor fragment will grow to form a mammary ductal structure, which, because the model that we transplanted in was null for p53, it would eventually develop mammary cancer. And so in this talk, I'm not going to go into detail of the effects, but but the foundations of so much of the research I do today began because of wrestling with this model. It's a great model, but it's also a very complicated model, right? And so my transition from being a molecular biologist to somebody who works with mouse models, right, that was it was non-trivial, right, as a mathematician would say, and quite a challenge. So one of the great features of this mouse model is that it produces tumor phenotypes or features of the tumors that are varied, similar to what we see in human breast cancer. And one example is the histological or histopathological subtypes of the tumors, right? And the three main types of tumors produced in this model are here on the screen: adenocarcinomas, squamous carcinomas, and spindle carcinomas. Furthermore, the tumors that were produced from this model can either be estrogen positive or negative for estrogen receptor alpha, which is a very important clinical marker in breast cancer. But more than that, the staining patterns of estrogen receptor alpha, right, were both canonical, right, on the left-hand side here, nuclear staining, but also non-canonical, that is, cytoplasmic staining. Now, these two slides right here, wrestling with complexity of that, okay, and understanding what it is that pathologists and biologists ignore because we can't understand, right, with our eye, with the naked eye, right? These two slides really would set the foundation on how I would later on, write computational algorithms to measure complexity of tissue architecture, right? Because if you look at the fact that a pathologist can actually look at a slide and say, yeah, this is an adenocarcinoma or not an adenocarcinoma, tells us that there are recurring patterns, right? Otherwise, the field of pathology wouldn't exist, right? But the limitation of the human eye is that, beyond these gross morphological features, it's really hard for us to assess, things that are more complicated. And here with the estrogen receptor alpha staining, right, it became clear to me as I was learning about estrogen receptor alpha that in the clinical setting, it's easy for the human eye to pick out, the degree of nuclear staining, right, as in the canonical pattern. But then in the clinic, there's just no practical way to assess cytoplasmic staining, right, as being biologically distinct from the nuclear staining, even though in basic science, we know that there are non-canonical signaling pathways for the estrogen receptor alpha, and that it does not need to be activated, by binding to hormone, but it can interact with the inner membrane of the cell. But in the clinic, a pathologist just wasn't able to assess that in a way that was practical, right? And so the wrestling with the complexity of histology and protein expression of clinically relevant markers really would set the foundation for the computational research program that I do today. So if I were to summarize the three main takeaways from my time at Mary Helen's lab, it would be these 3 points. That is, one, the biology of heterogeneity. That is, there is a science to understanding heterogeneity. And that, when scientists say that the system is heterogeneous, what they typically mean is, quote, I don't have a systematic way of measuring this, so I'm just going to ignore it, unquote. That's what heterogeneity means, right? At least in my experience in literature and talking to people. And in order to quantify, the disorder that, the order that we see, right, on a large morphological level, a gross morphological level, but also to quantify in the microscopic level, I needed to relearn my notion of randomness, right? This thing often used interchangeably with heterogeneity. And it turns out that there's no formal mathematical definition of randomness, right? Intuitively, we know what it is, but if you're trying to define it, scientifically, mathematically, it turns out nobody's able to do that because it's kind of very nuanced, right? And so I then realized that in order to actually write algorithms, I had to eliminate the word random from my vocabulary and just call something disordered, because if it's disordered, I can measure it precisely. But if it's random, like, I don't know how to measure it meaningfully in a scientific way. For example, imagine trying to collaborate with somebody who doesn't believe that there are 12 inches in a foot, right? And so and across different fields of science, turns out there's no one definition of what is random, especially in the computational sense when you have to define it. So that would be the first takeaway. Second takeaway is that in Mary Helen's lab, just looking at this complex in vivo data, and also doing a lot of gene expression analysis, that a lot of times we found patterns of enrichment of certain biological features like T cell activation or macrophage activation, and that the p-values, would suggest insignificance, but, these patterns would recur again and again, right? And such that intuitively as an experimental scientist, right, you have to think that, there is some significance, right, to it, this, despite the p-value. And turns out, it really made me think about what is the use of, computational studies and these things called p-values. And there's a difference between computational study standing on its own, right? An entire paper based on the computational studies versus computational studies as a tool to generate hypotheses that you can verify experimentally, right? And so that I'm not going to talk a lot today about the gene expression, lessons that I learned, but for those who are interested, I ended up writing a handbook on how to interpret gene or protein interaction networks, and you can get a free copy. Just email me. It's on Kindle Direct Publishing or Amazon Kindle. But really, this handbook came from wrestling with all the gene expression data we had, right, from our tumors. All right, and so moving on to the third takeaway lesson is that there is a tissue spatial code that governs development and tumor genesis, and we know that because the field of pathology exists, right? Again, the fact that a pathologist can look through a microscope and say this is a squamous carcinoma means that tumors do follow certain rules. Otherwise, pathologists would be out of jobs. And we also know that this code exists through regenerative medicine studies, right? Even our mammary gland transplantation, why the mammary gland constantly grows the way it does, right? And there are patterns, evidence of this code in many other animal models, which I will not go into, but certainly it was my time in Mary Helen's lab that solidified the idea that there is a code that we all know is operating. It's just nobody is able to articulate it. And so the goal of my lab is to develop first the computational tools to measure it. And then once we are able to measure it, we'll be able to crack the grammar, right, of this code. And once we crack the grammar, then we can figure out the syntax. All right. So I've developed a number of algorithms, but today I'm just going to talk about one of them. And this one's called the Quadrant Slope Index. And if you look at the panel on the left, oh, by the way, can somebody, Sylvan, somebody time me? I don't see a timer on my end. So in fact, I see it now. I'm already at 10 minutes. Okay. So let me quickly skip through this. That is, so on the panel on the right, panel A on the left, excuse me, is a cartoon of a tumor. And as you can see that, the spindle cells on the upper right-hand corner are obviously the human eye, but there's also a hidden layer of, a nuclear alignment on the bottom left-hand corner that the human eye is hard to pick out, right? So I developed a method to essentially highlight that. And the way to do that is to turn a tumor image into just a raster or a grid. And then in every grid, if you just measure the longest length of each nucleus, right, you get a line. And relative to the horizontal axis of the screen, you also get an angle. So if you just count up all the positive slopes and the negative slopes in each square, and then you divide it, positives over negatives, what you do is you turn each square into an intensity value, right, like a heat map. And when you do that, right, you're able to turn histology image. Here, these are examples of bladder tumors. You turn histology images into heat maps, right? Even though heat maps are great for visualization, they're even better for quantification, because you can turn the heat map, you can rank each of these intensity, the squares of these intensities, excuse me, the intensity of these squares, you can rank them from high to low and turn them into quartiles. And so when you can turn things into quartiles, then you can do statistics on essentially these heat maps. And when you do that, turns out you can stratify or quantify the difference between high grade and low grade bladder tumors. Okay, but more exciting than this is that if you look at the scatter of the high grade, what we call high grade bladder tumors, right, you can determine whether or not there are unidentified. For example, right, are the top quadrant of the scatter of high grade bladder tumors, are they enriched for certain features that are not enriched in the lower quartile? And indeed, we found 4 lines of evidence that this is the case. I'll quickly go through it here. First is that the fourth quartile, or the very bright green squares, okay, in high-grade tumors have a higher index of the ratio between GATA3 and cytokeratin 14, two very important markers in the luminal and basal compartment of epithelial tissues and tumors. And that there was also a difference in the very bright green squares, okay, in terms of tumors that had a lot of bright green squares. They also had a lot more nuclei per field of view, and the length of each nucleus was smaller. Lastly, I also looked at the different types of chromatin patterns in these tumors, and it turns out that the high-grade tumors, the top quartile, was enriched for a very distinct type of chromatin patterns, while the bottom quartile was enriched for a different type. And as an internal control, we looked at these chromatin patterns in low-grade tumors, and the low grades, majority of them were of just one type. And so the QSI algorithm, we've also been applying it now to melanoma and glioblastoma, and the results are very promising. And so as of now, there is no grading system for melanoma and glioblastoma. There are staging systems, but no grading system, probably because it's just so complicated for the eye to assess, all this complexity. However, in a matter of 12 to 18 months, I hope to falsify these statements in blue, because as our data is indicating, there are subgrades, and that we are in the process of clearly identifying them. And so this is that's the end of my talk, and here's just a summary again of 3 main takeaway lessons that I learned from Mary Helen's lab.
Thank you, David. I think you have a question from Alba. Hi, David, this is a really interesting tool. How would you view this algorithm in light of the current AI-powered image analysis tools that, for example, are being used in pathology?
Yeah, that's a great question. It certainly, it'll be another useful tool in digital pathology. So AI right now, the main tool for classifying images, whether it's tumors or whatnot, is convolutional neural networks, which is great. This is a complementary tool in that, convolutional neural networks is great for learning, training computer on complex things, but the problem is that is not always interpretable as what the computer is picking up. Furthermore, you need a lot of samples to train, right? At least 1,000 images per category. But my approach allows for a lot of user guidance, right? In terms of a biologist measuring specific things. And so what happens is that my algorithms, turns out, is like, according to my computer science colleagues, they say that's the old school way of doing things, but apparently it works better than convolutional neural networks in terms of finding differences with very small sample sizes, right? And it's because, convolutional neural networks were designed to require minimal user input and guidance. But as a biologist, I come to computation from the biology side, right? So I'm all about asking very specific questions. So the algorithms I develop turn out to be much more sensitive because it allows me to sort of front load a lot of information in terms of what question I'm asking. And so I would say that, my approach will help you identify subtypes that are unknown. And once you identify those subtypes, then you can train a convolutional neural network to quickly classify them, right? But my approach is much better at finding unknown subtypes, simply because it allows the user to put in front load a lot of sort of information in the question you're asking.
Dave, I think this is really, really fascinating, because what it demonstrates is really how combining biology with a physicist like Sylvain really shifted how we worked on a given set of problems that, so since my training came as a pathologist, I trained you in pathology, but you had the context of LBNL, which was highly computational, with Braun Pravin and Zhenhua Mao, whom you worked with over the course of the years. It's just fascinating how you actually demonstrate one of the principles I always ask, or I think is important in science, is what kind of information do you like to think about? You like to think about images. You see things, which is not a common trait, but being able to translate that into quantitation is really what I think is what radiation biology brings to biology, is that quantitative nature that comes from the physics side. I really like this, and we're going to have to talk some more. We have a lot of tumors to work on.
All right.